Petra, for the kind introduction, and thank you so much to the organizers for having us here. It's a great pleasure and honor to present at your fine conference here. Now the table moves up. Thank you so much. Okay, so in this little talk, I would like to give us all a break from AI. I will not talk about AI and 100% talk about BI. And with BI, I don't mean business intelligence, I mean biological intelligence. Because biological intelligence has this cool feature that AI doesn't have, I think, and this is called thinking. So let's talk about thinking. As Petra already introduced, we are doing very similar things with MRI data. We are pulling them through the usual pre-processing pipelines to extract the brain, the surfaces, surface reconstruction, region parcellation, diffusion-weighted MRI tractography, in order to then arrive at a simplified and aggregated brain model. So this is a large-scale brain model. The brain is parcellated into 380 regions roughly, and the connection strengths, the fiber bundle capacities between these regions is then reconstructed from MRI data in order to arrive at such a abstract model of the brain. So as input, as mentioned, we take diffusion MRI tractography and also structural data, and use this to arrive at such an aggregated description of the brain, and use the same space then with fMRI data to fit the model. So the model is based on structural data, and fMRI data is then used to fit the model. And the fitted model then is used to explain behavior. In this concrete study, there were 1,200 subjects from the Human Connectome Project Young Adult Study, which has, as you all know probably, high-quality MRI data, but also high-quality extensive behavioral testing. So we have intelligence tests of all these subjects, which is very nice to do research. We will go through several topics today, so if you are interested to read more and get to the depths, feel warmly invited to read our publication here. So what is intelligence? A really big problem studied by humans, at least as we know, at least from 1880. So since quite some time, we still don't really have a conclusive answer. Why do some people solve more problems than others? What distinguishes human intelligence from ChatGPT? Complex problem often is not like in AI, a direct mapping between some input and output, but it requires a recursive decomposition into easier subproblems, forming a hierarchy of decision-making processes. And this requires that all these subproblems higher up in the hierarchy need to be held in working memory until solutions from problems lower in the hierarchy arrive and can be integrated into the overall solution. So this is the opposite of jumping to conclusions or finding the direct answer based on an input pattern. And our theory is rather that true human biological intelligence is a scale-free fractal process that really happens on all scales and depends on multi-scale excitation-inhibition balance, because excitation-inhibition balance modifies the timing with which information is integrated to form a decision. We will look at this closer during the talk. So we can here really contrast two different forms of intelligence. So we have crystallized intelligence on the one hand, which can be learned. You can study it, you find the solution to the problem because it already exists. But the interesting problems are those where no solution exists already, and this is fluid intelligence. And it requires that the solution to the problem is figured out via thinking as the problem is worked on. And so this can be contrasted, for example, here. We have easy and fast problems that allow us to immediately come to a decision, like interpreting a traffic light. We can, if it's red, we can immediately hit the brake. But if we want to know how we get from here, from the Charité, maybe to Alexanderplatz, it really requires thinking. We need to look at this map and need to figure out the solution. And to test this, fluid intelligence tests use often Raven's progressive matrices. And they can also be easy and they can be hard. So for example, on the left side, just by looking at it, I think most of us immediately see the solution, right? These objects, so we have 3 rows with 3 types of objects, and they are just tilted 90 degrees to the right. We can see this just by looking at it. On the right-hand side, I don't know what the solution is. I really need to think about what patterns are shown here in order to arrive at the solution. So what underlies intelligence differences? And the classical theory since 1880 basically is that smart people are smarter because their brains are faster, in a very hand-wavy fashion formulated, of course. But this is really an influential and strongly supported theory. So this was over the decades now proven over and over again, really with tens of thousands of participants. And this is a very simple test. Click a button, hit a button when a certain number or color appears, and don't hit it when another color appears. Takes a few milliseconds. But nevertheless, it gives remarkable information about the physiological state of the subject. So this processing speed, this simple metric, correlates so tightly with most other intelligence metrics. And also the decline of intelligence over lifetime is captured by this metric. Also, it's famously used as a better explanation or predictor of impeding death than the usual suspects like smoking, physical activity, resting heart rate, blood pressure, alcohol, and so on. So there's really something about processing speed that really relates to our physiology, at our bodily system integrity, and maybe to our efficiency of information processing. But is processing speed really the causal factor for intelligence? So one important counter-argument is that training processing speed doesn't make you smarter. So it doesn't transfer to other cognitive domains. Also, you can measure that gray matter changes and so on. Cognitive function doesn't really improve by training your processing speed. You cannot really train for fluid intelligence. With complex problem solving, the timing really matters, as I alluded to earlier. The goal is to infer a hidden rule that is yet unknown. So the strategy is a recursive decomposition into simpler subproblems, forming a hierarchy of decision-making processes and problems. And this requires then making decisions about tentative solution paths while storing the previous results in working memory and integrating them all upwards again into the hierarchy. And of course, the hierarchy needs to be held somewhere, either in working memory, then it's usable, or somewhere stored in synapses maybe, but then it's not really usable. So it must be there to process and to arrive at a solution for a tough problem. And this is something that was also called System 1 and System 2 thinking, for example, by Daniel Kahneman, who unfortunately died last year, but received a Nobel Prize for exactly this theory. So we have a System 1 that operates very quickly with little to no effort, and we have a System 2 that really requires the allocation of attention to an effortful mental activity, and is often associated with agency and choice. And mistakes occur if we allow our System 1 to make a decision where actually System 2 should have done it. So we now recast this influential theory, thinking fast and slow, into decision-making fast and slow. So we bring it onto a mechanistic brain network level. And we actually found that participants with higher intelligence were only quicker when responding to simpler problems. What no one seemed to saw before is that they needed more time to solve the complex problems. So you can see this pattern clearly in the empirical data. No neuroimaging required so far. This becomes apparent in the Raven's matrices test because the 24 questions of this test, like I shown earlier, they are sorted according to difficulty. So we start with the easier, and it gets harder and harder and harder, and there's no time limit. The task is think until you find the solution. And if you cannot find the solution anymore, then it stops, or if you found a wrong solution. So these tests can be unsolvable, even without time limit. So the trivial explanation is that, maybe since they are sorted in order of increasing difficulty, and they're discontinued after 5 incorrect answers, people could have a higher reaction time, so the higher intelligence people could have a higher reaction time for more complex problems simply because they advanced further in the test. So let's look at the individual questions. So at the top, we have the simplest question, question number 1, and the hardest question, question 24. At the x-axis, we have the G factor, the intelligence of the subjects, and at the y-axis, the reaction time. And so this is at the top group average. At the bottom, then on the right, we see individual subjects' correlations. So we clearly see for the simple questions, the smarter subjects that come farther in this, that have a higher intelligence according to these tests, they are quicker to solve the easy problems, but then there's a turning point where they get slower. Another thing we found then when we correlated the reaction times of the individual fluid intelligence test questions with the fMRI data, specifically the functional connectivity, is that we always have this strong correlation between reaction time and functional connectivity independent of intelligence. So there seems to be higher functional connectivity doesn't seem to make you more intelligent per se, but it seems to have something to do with when a choice is being made and how people work towards a solution. So now we want to study this mechanistically. And to do this, we also did a thing that seemingly no one did before. We added feed-forward inhibition. So normally in brain network models, you have excitatory populations, inhibitory populations, and they are locally coupled with each other. But on the large scale, it's only the excitatory populations that are coupled with each other. There's no arrow so far going from excitatory to inhibitory populations. So we added this because this is what exists in real brains, but wasn't simulated explicitly so far. And this now really allows us to tune excitation or to control and tune excitation-inhibition balance. So on the left-hand side, we see how a high excitation-inhibition ratio would look like. There's a strong connectivity between the excitatory populations and not so much from the excitatory to the inhibitory populations. On the right-hand side, we see there's not much connectivity between the excitatory populations, but a lot going to the inhibitory populations. So this is a low EI ratio. So we can formulate the EI ratio as the ratio of long-range excitation to feed-forward inhibition. So feed-forward inhibition, there's an excitatory synapse or synapses going to the inhibitory pool, and then from the inhibitory pool, the inhibitory synapses go to the excitatory pool at the other brain region. And by tuning now EI ratio from low to high, we can actually tune the functional connectivity between these two brain areas. So we now start the video, and we start on the very left-hand side with a very low EI ratio and go then towards a high EI ratio. You can smoothly tune, it's the same noise that was input, you can smoothly tune the correlation, the functional connectivity between these brain areas. We can basically now smoothly tune synchrony from anti-synchronization to synchronization via a critical, complex, and intermediate state. So here we can see the extremes again. So on the bottom, we have synchronization. So you can see the high FC between these brain regions. They do more or less the same. At the top, we have the exact opposite. The one brain region does this, the other does the opposite. And now by tuning, you can have everything in between, right? And have also this complex critical state in between where, which looks very chaotic. So we are pretty convinced, I at least, that this could be a fundamental principle, even down to the quantum scale, because we have excitation-inhibition balance everywhere. We have some in neurons, obviously, but also if we go down the scales, in metabolic pathways, in metabolic networks, we have upregulation and downregulation. On the level of atoms, we have excitatory and inhibitory parts. So you can use this principle for everything. So I would love to program a quantum computer and really build models from the bottom up based on excitation-inhibition balance. And this doesn't not only work between two populations. Let's do the same now for every population in the brain. And yeah, it doesn't depend on how many regions we have, how fine the parcellation is, on which scale we are looking at. We can just yield a very simple algorithm based on this simple principle that acceleration-inhibition balance has a smooth relationship with functional connectivity. So when we see that functional connectivity between any pair of brain areas is too low, we simply increase the EI balance. If it's too high, we decrease it. That's it. So we can do this now for all 380 regions. On the left-hand side, you see our goal, the empirical FC, and on the right-hand side, the simulated FC as we start the simulation. Now we start the tuning process. So let's now fit the entire brain model. As you can see, somehow it can be tuned to perfect correspondence, basically. So before that, a brain model would usually have an FC correlation of, let's say, 0.5, 0.6. And now you can tune any network to any functional connectivity if there are excitatory and inhibitory elements in a network. So I think this pretty much now links, actually, correlation and causation. So what does this do with the intelligence of the people? So now we, after fitting every model to every single subject of the HCP data set, we looked in the model what is different, what is specific about those subjects that yield a higher IQ on the intelligence test versus the others. And two things were striking. The synaptic input current amplitude was lower for the high IQ people, and the synaptic current correlation was higher for the high IQ people. And now we would like to test what happens in classical decision-making models, winner-take-all decision-making, like these four competing populations here, when we drive them with exactly such a pattern that we found in the subject-specific models. So we implanted this winner-take-all decision-making circuit into posterior parietal cortex and prefrontal cortex and used the large-scale brain network to drive the winner-take-all mechanism. So this is there's cross-inhibition. Two populations are fueled with evidence, and the more evidence then is the winner and takes the decision. So in the empirical data, we found that the slower subjects found a better solution and that they had a higher FC. In our model then, we found, very roughly speaking, the details are much more complex. Please read the paper if you want to learn them. We don't have the time, unfortunately. Then in a large-scale model, a higher FC was accompanied by a decreased amplitude and an increased synchrony. Now, what happens when we test exactly this in the small-scale model? We find that a decreased amplitude leads to increased synchrony, and a decreased amplitude and increased synchrony leads to slower but better solutions. And now, by looking at the phase space of the winner-take-it-all model, we can actually also see what mechanistically happens when people slow down to integrate more evidence to form better decisions. So on the x-axis of the phase space on the right-hand side, we have population A. So this is decision for option A, and on the y-axis we have population B. And we can see here, this video then will start in a state where there's more amplitude going into the population. So this is more like the jumping to conclusions, the not thinking at all, but just taking whatever answer is there. And this is accompanied by a fixed point that is very up in the state space and directly pulls the populations active, and then with the next best evidence coming, draws it to one conclusion. And when we now decrease the amplitude, we will find that at a lower point in the phase space, a new fixed point will emerge that keeps both populations in a slightly active state, like the tennis player being alert of the incoming ball, serve, however it's called, and awaiting the reaction, basically. So let's start the video. So we can see when the amplitude decreases, then a new fixed point is generated at the bottom, which allows to keep populations active and integrate evidence for a longer time. The same thing we studied also with correlated input activity. So how similar is the everything else that is not evidence that goes into population A and B. And for this, we have these stochastic phase portraits where these glyphs, these round objects, denote basically all flow that is independent of the actual evidence, but that comes from the brain network background. So every other input, these populations that are supposed to make a decision now for a certain concrete scenario get also, and it's kind of distracting for them. So here we have basically shown that with higher correlation, the flow is more bound towards the center diagonal and also keeps the decision open for a longer time and not directly jumps to a conclusion. A very similar thing, so with these winner-take-all models, you can also study working memory as persistent activity, and here also the same needed pattern emerges, that the decreased amplitude makes the working memory more robust and less likely to override a current working memory state, while an increased amplitude makes the working memory more responsive to new stimuli when suddenly the traffic light shifts to red. So now we have the large-scale models. They were fitted to each individual FC of each of the subjects. We have implanted the winner-take-all model in PPC and PFC of each of these subjects. And the brain models then did what the actual persons did. When they took more time to form a decision, they made less errors. So as a summary of this first part of the talk, intelligent people take more time to solve complex problems, but are faster to solve simple problems. Brain network models can be fitted with arbitrary functional networks based on a biologically plausible fitting algorithm, namely excitation-inhibition balance. The dynamics of the fitted models relate to cognition of the subjects, supporting inference of healthy and pathological neurocomputational mechanisms. And by coupling decision-making and working memory circuits with brain network models, we can now simulate high-level cognitive processes, task virtual twins. So as a step to artificial human intelligence. And we see here now very clearly also what already Daniel Kahneman alluded to, that also in the brain there's no free lunch. There's always a trade-off between speed and depth of information processing. And so in a 2025 Neuroscience talk, we also need to talk a little bit about science operations and data protection and how to work with data and data infrastructures. And we are happy to collaborate here with a team which is now extending what comes from software engineering towards better science operations. So from ad hoc processes, from try and error experiments, we go over to repeatable processes, towards open source ecosystems, towards automated pipelines, and ultimately AI and human in the loop. This is what everyone's doing. One can clearly see also in this consortium, this is the typical hierarchy of how science operations scale up. This is what we, of course, also wish for our platforms. So we use automation, continuous integration, and continuous delivery pipelines, shared code bases and version control for better collaboration, tested, monitored, and repeatable systems in order for better reproducibility, scalability, security, and compliance. So what inspiration can we take from DevOps? Git workflows, of course, for version controlling code and data, thanks to data-led, and CI/CD pipelines, so always having the full pipeline intact, and when some upstream component changes, immediately rerun all analysis to see whether this has any impact on the result. infrastructure as code, the whole platform, the whole tech stack basically from the infrastructure up to all applications should be reproducible and not to not run again into the old problems that some dependency was just not there, making results not reproducible. And like everyone else, we are using Docker containers for that and orchestrate them with Kubernetes and then use Terraform to specify the exact state of such a cluster and make it also automatically deployable. Of course, we try to modularize every component into microservices so that they can be easily reused and combined and used together with monitoring and observability tools. This is, of course, to help science, but related to all this technical work is, of course, also the protection of the privacy of the data that we have here. As you see, these are very personal things that we are analyzing here from the subjects that donated their data to science. As you all know, we have fingerprints all over our bodies, so not just at the tip of our fingers, but every voxel basically in an MRI could be a fingerprint. This was shown. Even with very incomplete and very small data sets, you can then at some point re-identify humans. For example, in the Human Genome Project, which sequenced the first human genome, it was possible to re-identify the subjects based on publicly available genetic data from these companies that send these home test kits for DNA, which is, of course, a big problem. To work on this, we create the VRE and the Health Data Cloud, which are neuroinformatics platforms for the secure processing of health data. So these are data management cloud platforms for biomedical research that store, process, and share data in compliance with GDPR. It provides graphical user interfaces, command line interfaces, and secured, isolated spaces for private cloud computing. So your team gets virtual machines where you can install all the software and use it to analyze your data in security. So typical user-facing functionalities of a data management platform, a web GUI, a command line client, Jupyter Lab, and also an HPC connection. All of this based on open source components and scalable as in a cloud ecosystem. So it, as mentioned before, makes use of all the typical technologies for cloud operation. And of course, ensures that the data is secure. So, for example, the data center is in the Charité, which is a critical infrastructure under the German law with very high security requirements. And isolation, of course, firewalls, data zones, encryption, authentication, authorization, and access control are widely used here. The whole system is also available in an automated deployment fashion, so you can relatively easily deploy the system at different sites, and this is then also our goal to scale up the system as a network of different sites that allow to exchange data, or at least metadata, and expose metadata in a compliant and secure manner. And with this, I'm at the end, and thank you very much for your attention, and I'm happy to take questions.